I just want to finish this up about water, right, and how it relates to this acids and bases and salts, right? So anyway, we want to look at these other kind of solutions, things that dissolve in water, namely acids and bases and salts, right? So the salt, right, like sodium chloride or something, like right here, put it into water and it dissolves into its constitutive ions over here. And that'll be true for, potassium hydroxide or hydrochloric or hydrochloride, right? If you take those, they'll dissolve into their constitutive ions, right? The difference here between acids, bases, and salts is these, if it has a hydrogen or a hydroxide group, it's an acid or a base, right? Versus a salt. If it doesn't have that, it's a salt, right? Because these hydrogen ions are going to be the main player in our pH thing. And these hydrogen molecules, how acid a water is, how the level of acidity in the water is going to play an important role in all these chemical reactions, right? So these are, even though they're kind of dissolving ions, right? The fact that there's a hydrogen or a hydroxide ion is going to make this an acid or a base, respectively. Right? So they're both dissolving into ions, acid, bases, and salts. But these, if you see a hydrogen or a hydroxyl group, then it's an acid for the hydrogen or a hydroxide for a base. So what this relates to is the pH, the idea of pH, right? And this all relates to the amount of hydrogen ions in a particular solution. So this scale is a little confusing to some people because we say as a solution gets increasingly acidic, the pH gets lower. So a solution with a very low pH has more acidic than a solution with a higher pH, right? We start at number 7 here as a neutral, and the highest pH you can get is at pH 0. And so the reason for this is these numbers over here are actually reflecting these numbers over here, the concentration of hydrogen ions. So this 10 to the 0 right here actually means 1 mole per liter. This is 1, we'll just say M solution here, whereas this one with a -1 means 0.1 M because we've moved the decimal point over one space. If we move it over 2 spaces, we have this 10 to the -2. So each of these is a decreasing concentration of the hydrogen ion. And so these numbers are just there reflecting these actual concentrations, the molarity of the hydrogen ions, right? It's actually a little bit easier, although, as I said, it is a little bit confusing to say you're getting an increasingly acidic solution and it's the pH is lowering. So the other thing to realize about it is that for each numerical change in the pH going either direction, it's a 10-fold change because this means 1/10 of a mole. This means 1/100th of a mole. This means 1/1000th of a mole. And this means 10,000ths of a mole. So any change in a whole number of this pH is a rather drastic change in the actual acidity. So our blood is at something like pH 7.35 to 7.45, right? A fraction of one of these whole numbers, right? Because the change from this to this is very drastic. So your blood, for instance, is kept at within the range of pH of 7.35 to 7.45, right? Because this looks like a small difference, but again, each one of these is a 10-fold difference, right? So the blood must be kept within a certain pH range, a condition of acidosis occurs. If it falls below that, if it goes above that, you are in alkalosis. So Acidosis and alkalosis contribute to a wide range of diseases, and the mechanisms are complicated. But one easily demonstrated harmful effect of low pH is how it disrupts enzyme function. We talked about that protein denaturation, right? If their pH was too low, right? If there's too many acids, the protein loses its shape, right? And it doesn't do its job right over here, right? And this is because, these proteins have, remember those hydrophobic interactions. It has hydrogen bonds. It has these disulfide bridges, right? And if you add too many hydrogen molecules in here, they start interacting, in particular in this case with those sulfide bonds, these disulfide bonds, and they break apart the shape right here. So instead of these sulfur molecules binding with each other, they start to get these hydrogen attached to them and they dissociate and it becomes denatured, these proteins. So that's one way having a low pH messes up these proteins, which depend on their shape to do their job. Right. And so having this, not only just directly that pH, but you'll see these clinically. If your blood is too acidic, it's considered you have acidosis, right? And you have all these different symptoms which are related to having too many free hydrogen ions in your body, right? If your blood becomes too basic, that is not enough hydrogen, you also have these other types of problems right here. So alkalosis, we'll run into these, and you'll certainly run into those when you get into the next Bio 145. Right? So this is one of the main problems with this acidity of the blood, right? Acidosis, alkalosis, too many hydrogen oxygens in the blood. So in order to maintain, right, the proper pH, right, we have to have there have to be buffers, right? Buffer solution. The blood has to be some kind of a buffer, and what that means is buffers are going to stabilize the pH by either adding or removing protons, right? So we want to look at these reactions here. Right? And basically the idea is that a strong acid like hydrochloric acid, you put it in there like a salt, will completely dissociate into its constitutive ions. If you have a weak acid, over here, you put it in there and it's only going to partially dissolve right here, right? So this is the difference between a strong and weak acid. And these weak acids over here, usually denoted by this HA, right? It's going to act as a buffer, right? Because it's a weak acid, it only partially dissociates. So here is one major buffer system in your blood, which I've mentioned a couple of times right here. It's the most important one, really, that you'll keep on going over and over again because it's really central to a lot of processes, right? Most important extracellular buffer system in your fluid as well as in your blood, right? Remember that carbonic acid that was formed? Well, that's a weak acid that dissociates very quickly into hydrogen ions and this bicarbonate ion, right? Once you get into the physio stuff, you'll just call this bicarb, right? Because it's the very most common thing right here, right? So all this is a reversible action, right? And you could let off a couple of hydrogens very weakly, but you could also accept acids very weakly, right? So this is a very important buffer, in this case, this bicarbonate ions. So in this case, right, your CO2, which was not really carried well, was converted into this carbonic acid. It quickly dissociates into these two. So all your carbon dioxide is being, not all of it, but most of it's being carried around in this form and in this form right here. But so it's not only carrying around, it's acting as a buffer that's going to soak up extra hydrogen. This whole reaction is what we want to look at, this dissociation from carbonic acid to bicarbonate ion and this hydrogen atom right here. And on the other side of it, the main point of that was this reaction, the amount of carbonic acid in here is going to be related to the amount of carbon dioxide in the bodily fluids right here. So the more CO2 you have in the blood, the more this reaction is going to go this way. Because remember, this is a reversible action over here, right? So this formation is a way to carry this, but it's also going to soak up the CO2, make it into that, right? So the problem with this is once you get this formed, it dissociates into this. So the more CO2 you have in the body, the more the reaction is going to be driven this way, the more it's going to dissociate. So the more hydrogen ions you have in your body. So this is going to make a lot of hydrogen atoms, right? The more of that you have. The more carbon dioxide you have, the more hydrogen ions you have. So you got to find a way to get rid of this carbon dioxide so that the reaction is actually going to the formation this way. Right? So this is, the exhalation of carbon dioxide is one way to control pH in your body, right? So in your blood, some of your systems right here have receptors that are going to be able to detect levels of acidity in your blood, right? In your brain and your blood vessels. Your heart is going to detect that the levels of hydrogen is too high. They're going to send messages to a control center in your brainstem, which is going to tell you to breathe faster. When you breathe faster, you're getting rid of more carbon dioxide. And the more you get rid of carbon dioxide, the more the reaction goes this way, over here, which means there's less hydrogen ions formed, right? So you're going to soak that up. You're going to reduce the carbon dioxide. You're going to reduce the amount of hydrogen, leave more of this to soak up more hydrogen. And so the bottom line is this response, less carbonic acid forms, fewer hydrogens are present, and the blood pH is going to increase. That is, the hydrogen concentration is going to decrease. All right. So the other buffer systems in the body are, this is the main one we're really talking about. Carbon, it's called a volatile acid because it's going to be exchanged with the air, right? Whereas these other ones are always stopped in the fluid, right? These phosphate buffer systems, mainly inside the cell. There's also protein buffer systems, mainly, this is probably the main intercellular one where the proteins are going to act as buffers themselves, right? And taking up hydrogen ions. So these are the buffer systems that are maintaining the pH homeostasis. Last one, and I'm done here. Last thing about controlling these hydrogen molecules, the pH levels, is through the kidney, right? Through these different kind of reactions. Again, it's going to be involved this bicarbonate, where the hydrogen ions are going to be pumped out into the tubule to be excreted, right, with the rest of the urine. That's why your urine is acidic, right? Much more acidic than your blood or water, right? So there's another way that your body is controlling through your kidneys, and you learn all about that during your renal sort of physiology when you get to that portion next class. Not next class, but next 145. All right.